Assalamualaikum and hi. My name is Muhammad Uzair bin Tamizi from Group AT24002A. Today I will be presenting my case study for crop disease management on a severe threat facing our agriculture sector, Fusarium wilt in oil palm plantations. In this short presentation, I will break down the identity of the pathogen, the symptoms we look for in the field, its severe physiological impact on the palm tree, and the integrated strategies we use to manage it. Okay, let's move on to slide 2 and discuss what exactly causes this disease. The causal agent is Fusarium oxysporum FSP elidis, commonly abbreviated as FOE. This is a highly specialized soil-borne fungal pathogen. What makes FOE exceptionally dangerous in a plantation setting is its production of chlamydospores. These are thick-walled survival spores that allow the fungus to remain dormant in the soil or within all crop debris for decades. Even without a live host plant present, once active, it specifically targets the root system of the oil palm and aggressively colonizes the plant's internal vascular pipeline. Turning to slide 3. How do we recognize this out in the field? Early signs appear on the lower leaves showing interveinal chlorosis, which quickly turns into drying necrosis. As you can see in the field image on the slide, a classic symptom is the fracturing of the dead fronds. The dry lower fronds break near the base of petiole and hang straight down around the trunk, forming a characteristic skirt around the palm. In acute cases, especially with younger palms, the entire canopy can desiccate and turn yellow within weeks, drastically stunting growth and destroying bunch production. Now on slide 4, let's differentiate the structural signs of pathogen. Unlike some diseases, fungal signs are rarely visible on the exterior of a live palm. Instead, we have to look internally. Dissection of an affected trunk reveals an obsolete diagnostic signs, severe dark brown or reddish black discoloration of the internal vascular strands. Under a microscope, we verify the infection by identifying two types of spores: tiny single-cell microconodia inside the vascular tissue and larger signature sickle-shaped multiseptate microconidia after culturing the sample in the laboratory. On slide 5, let's go to slide 5. On slide 5, let's examine how this disease damages the core biological functions of the palm. Because FOE travels strictly through the vascular network, its hyphae, microconidia, and fungal toxins lead directly to vascular occlusion, a massive physical blockage inside the water transporting xylem vessels. The palm actually attempts to defend itself by producing gel-like plugs called tyloses, but this inevitably seals off its own water flow. Consequently, upward translocation of water and minerals is completely restricted. Cellular turgor pressure collapses, and the tree effectively dies of structural dehydration and starvation from the inside out. Finally, on slide 6, how do we control this aggressive disease? We must use an integrated approach. First, prevention relies on planting resistant oil palm progenies, particularly tested Tenera lines. Second, excellent field sanitation and the clean clearing of infected debris during replanting are required. Crucially, as we explored in our topic 6, lab activities on biological control, we can inoculate nursery soil with beneficial Trichoderma species. Trichoderma acts as an aggressive competitor that colonizes the root zone, effectively blocking out harmful FOE through competitive exclusion. Lastly, we optimize plant immunity by applying high potassium fertilizers to strengthen tissue resilience. That concludes my presentation. Thank you very much for your time.